Interesting information to cover today. I'm Mike Osterholm, Regents Professor and Director of the Center for Infectious Research and Policy, as well as a member of the Executive Committee for the University's Consortium on Law and Values. The lecture series is jointly presented today by the University of Minnesota's Consortium on Law and Values in Health and Environment and Life Sciences, the Center for Infectious Research and Policy, the Institute on the Environment, and Food Protection and Defense Institute. I'd like to thank the Planning Committee, which includes Louis Gilbert from the Institute on the Environment, Amy Kirshner from the Food Protection and Defense Institute, Susan Wolf, who chairs the consortium, who's in the audience, who we all absolutely owe all of this to for her tireless work in making the consortium happen, and I happen to be on it too. I'm the caboose. The consortium is a university-wide center that links 20 leading university centers and programs to address cutting-edge societal issues posed by biomedicine and the life sciences. I want to thank the consortium staff for managing the logistics of the lecture series, both today and the ones we have in the future. Our speaker today is Professor Andrew Dobson from Princeton University to speak on the dynamics and evolution of emerging pathogens, insights from California's salt marshes, house finches, and Zika viruses. I'll introduce him formally in a few minutes. Professor Dobson will speak for approximately 40 minutes. We will then hear from a faculty commentary from Professor Jessica Hellmann, who directs the Institute on the Environment. I will introduce Professor Hellmann briefly before she speaks for about 7 to 10 minutes. In terms of the audience, I want to ask all of you at this time that to keep your questions handy. We will have a time for those at the end of the presentation. There are 2 microphones here and here. We ask that you use either one of those microphones when coming to ask a question. This particular lecture series is being videotaped and will be simultaneously webcast, so we also ask that you not walk across the front of the stage to block the camera and to make sure that your questions can be heard to all the audience. I ask all of you to please silence your cell phones and all the other electronic devices. Anybody's cell phone who goes off during this talk will be responsible for picking up the bill for lunch. If you need to exit the room during the event, please use the doors to the rear of the room, not the doors near the stage here, over here, where you'll get in the way, actually, of the videotaping. To get continuing medical education or continuing legal education credits, you need to complete and submit a participation form at the registration tables just outside the entrance to the room. Other healthcare professionals can submit a statement of participation to their appropriate crediting organization or state board for consideration of credit. An evaluation form will be emailed to you right after this event. Please be sure to respond. We do pay very careful attention to this, and we listen to your feedback. In terms of disclosures, I want to make sure that you're aware of that. Copies of all disclosure statements are at the registration desk for those who wish to review them. Our speakers and planning committee do not have any relevant disclosures to announce today, so good reading. Now it's my honor to introduce Professor Dobson. I can tell you, at the very, very least, besides being a well-known and well-published author and professor, he also is a lot of fun to have dinner with wonderful conversation. Andrew Dobson is professor in the Department of Ecology and Evolutionary Biology at Princeton University, external faculty at the Santa Fe Institute in New Mexico, and the A.D. White Visiting Professor at Cornell University. He received his doctorate in philosophy from the Department of Zoology at Oxford University. Professor Dobson's research focuses on the population dynamics of parasites and pathogens in the Serengeti, Yellowstone, the coast of California, the Canadian Arctic, and the backyards of New England. I'm in the wrong business. I never get to go to those places. He also works on the interaction between climate variability and the transmission of malaria and cholera in India and Bangladesh. His research is sponsored by the NIH, the NSF, and NOAA. Each study focuses on different aspects of interactions between pathogens and their hosts. This has allowed him to develop a larger body of theory dealing with the role of infectious diseases in natural populations and communities. Professor Dobson is an elected fellow of the American Academy for the Advancement of Sciences and the Ecological Society of America. He is widely published, including over 200 papers and books. It's my distinct honor to welcome to the podium Dr. Andrew Dobson.
Great. Thank you very much. Thank you. All right. Can I start off by just saying it's a great honor to be here. when I was a graduate student, which was like before the internet, almost before computers, I used to go to the library and write these things called reprint request cards, when you wrote to somebody and said, I read your paper, I've seen your papers come out, can you send me a copy? And I was reminded when I got here just how many of those used to come to the biology department of the University of Minnesota, going to people like Margaret Davis, Dave Tillman, Craig Packer. So many, many people here influenced me as those envelopes appeared and I read through their papers. Now I just have to download the PDFs and not read them, but in those days, your department had a huge influence on what I do. Okay, so it's a great honor to be here. What I'm gonna do is give you a talk that sort of initially says what I don't think we should do about emerging diseases, a little bit of a critique, maybe, of some of the talks that have come, partly to say what should we be doing. And to do that, I'm going to give you 3 little vignettes that try and put that problem in perspective, and then a longer, detailed look at one emerging disease, which is one of the best ones to study because nobody cares about it. And if nobody cares about it, we're not trying to get rid of it, so we can see what happens to an emerging disease as it evolves and turns into a new disease. So there'll be 3 vignettes, a little bit of a rant at the beginning, and then a more detailed study. Now, hopefully this will go forward. Okay, so how worried should we be about emerging diseases? It depends who you ask. If you go to Hollywood, they want you to curl up and die, although if you're lucky, they'll make a vaccine by next week and we'll all be saved. And the general theme of the Hollywood movies on emerging diseases is that. If you're in Washington, it's okay. God has sent these diseases to punish you, unless you're one of them, so just take your medicine. If you're some of the Predict EcoHealth Alliance, you're predicting there are millions of those viruses out there, so give them lots of millions to make maps of this, and that'll save us. The real line that I'll get to is Mark Woolhouse's graph, which says, actually, we get about 2 new viral diseases in humans every year. So let's just look at that. One approach would be to make these maps. And these maps tell you, or supposedly predict, where you're going to get new disease outbreaks. However, if you look at them closely, they actually tell you where there are research centers working on emerging diseases. People can then use these maps to make even more detailed maps of where are the hotspots for emergence. But the thing I worry most about these maps is these are the data they're based on. They're very pleasing to the eye, but sort of beware of false prophets. If you look at the underlying data, it's these 200 data points. So making a predictive map out of 200 data points is a bit like trying to predict the election based on 1,000 data points, and we know how useful an exercise that was. So the mapping software is incredibly powerful, but the pictures you get are only as good as the data goes into it. And this data, Mark has analyzed, this is data from Mark Woolhouse. He's analyzed that very carefully and essentially shown, if you look at emerging diseases, going back to when we first knew there were such things as viruses, so this is sort of measles and mumps, there's been a steady slope that suggests we get 2 new viruses in viral things that could be pathogens in humans every 2 new viruses a year. We can see some notable dates. Zika appears initially in 1964, but we didn't really start worrying about it until last year. HIV-1 appears in about 1983. HIV-2 we recognize 86. That was the most scary emerging disease we've ever seen. And then we had Sars, which caused huge economic upsets, but mainly only killed a couple of hundred people. You can actually look at that in terms of the number of viral families and see we're actually beginning to steadily satiate the total numbers of viral families that are out there. So what I'm going to say is it worth doing all this virus hunting? How, and in many ways, how similar is hunting for new viruses to stamp collecting? The better class of questions we should be asking is how unusual a pathogen jumps between species. What are the mechanisms that cause that? And is it naive to think that undescribed viruses are restricted to the exotic tropics? So to address those questions, I'm going to take you on a little trip round a bunch of research projects. The majority of these have been funded out of the NSF Ecology of Infectious Disease program. We're going to start off going to California. We're going to look at this question, how many viruses are there out there? just by looking at one ecosystem. We'll then go to the Serengeti to look at how do we control these diseases? When do we start seeing unexpected effects that we wouldn't see unless we used mathematics to look at some of these patterns? We'll then go to Brazil, home of the World Cup, and things like that. But there we'll focus on the very scary outbreak of Zika that occurred last year. And finally, for the longer section, we'll initially start at the Jersey Shore, my home base, and then look across the whole US at an emerging pathogen there. The underlying themes are how much pathogen diversity is there, and what's the interaction between population dynamics of the pathogens and host immunity in determining what happens to pathogens as they emerge. So let's go to one of my favorite places. This is Carpinteria Salt Marsh. It's just south of Santa Barbara in California. It's on the coast of California, and with a wonderful group of people based around Kevin Lafferty, Armand Curis, Ryan Hettinger, what we've been doing is dissecting these salt marshes to say how many parasites are in there and how many free-living species are in there. Essentially, what we do is we divide the salt marsh up into different areas. We go in with a large field crew, 2 sets of 20 people, a vertebrate crew and an invertebrate crew. We sample in quantitative ways the salt marsh to get to the diversity, abundance, and biomass of everything in it. And we've replicated this at salt marshes all along the coast of California. We use really subtle techniques to see what's going on. We collect this dominant snail species. We hit it with this very finely structured dissection instrument, having measured it, and open it up to see 2 organisms. The front end of this, which is this end, is a snail. The back end is all tissue that's been converted into a parasite to produce parasite-infected stages, Cercaria of a trematode. So we can measure half the weight of this as snail, half of it as parasite. Its phenotype is that of a snail. It's sterile, so its genotype, it's only producing parasites. It's been taken over by an alien species, if you like. It produces these wonderful things, Digenaean Cercaria, which if you went collecting them without your welly boots on, you'd start scratching a bit because they're trying to colonize you. It'll give you a nice little dose of swimmer's itch, but it's not going to infect you. What it's looking for is a fish. It goes into the fish. It forms these minute cysts on the brain, changes the behavior of the fish, and makes it easier for a seabird or seagull to eat that fish, where the adult parasite then develops in the guts of the seabird. So you have a parasite that has 3 or 4 different morphological forms, all living in different trophic levels. Now what we've done is painstakingly, in 3 different marshes, look at the biomass of all the different host species, the proportion of them that are infected, and what's the weight of parasites in this. So this is on a log scale. These bars here are the birds in 3 different marshes, 1 in California, 2 down in Mexico. So on average, the biomass of birds is about 5 to 8 kilograms per hectare. If we then compare that, converting the parasites up to biomass of the parasites, you can see that in these marshes, the biomass of some of the parasite stages is more than that of the birds. There is a huge biomass of parasites in this ecosystem. It's just hugely distributed in tiny little particles. And indeed, one of the tasks has been to put together a food web. This looks like a complicated knitting diagram, but essentially it's the number of species on each different trophic level and how they're connected together. That's the 6 different trophic levels of free living species. It's plainly a complex problem. When you put the parasites in, the whole system is even more coupled together. And it straight away tells you there's a huge diversity of parasites in that marsh. In fact, there are as many parasitic species as there are free living species. We just don't have enough taxonomists to identify all the parasitic species there. So there's a huge diversity of species. Again, we can do that in fancy 3D. This is the free living web. This is the parasitic web. There's a huge abundance of parasitic species connecting all the free living species together. And that means if we go into that marsh, we're going to find lots of new species. Particularly, these are only the things we can see under a microscope. If we go down into the sort of genetic level, there's about 1,000 species of parasitic things in there, which I can show you. Now, how are they distributed? Well, if we look at this by trophic level, If you're at the bottom of the food chain, you don't have many parasites. The higher up the food chain you go, you've got more and more parasitic species. So by the time you're at the top of the food chain, you're essentially dominated by things eating you from the inside out rather than the outside in. That's actually quite good news for what's going on in Washington. The higher up you are in the food chain, the more parasites you're going to have. As I say, if you then Forest Rowers Group are looking at similar marshes down in San Diego. If you go into the mud and just start looking at the microbiome, you see exactly the same thing. There's about 1,000 species of things with RNA and DNA. Roughly half of those are parasitic on the other half. So in these salt marshes, there's huge numbers of undiscovered viruses. We don't know what they do. Maybe some of them will jump over into humans, but I bet if you go in your backyard, you're going to find similar patterns to this. There is a huge viral and bacterial diversity scattered around the world. I don't think we're ever going to be able to really do the taxonomy on all of it. One nice thing, if you take all these things, just dividing up into what's the relative body size, you have as many things eating you that are bigger than you, those are predators, than eating you that are smaller than you. If you look at the biomass of everything, you can essentially show that as you get bigger, you're less and less abundant, but the smallest things are distributed in abundance using roughly the same scaling laws as the larger things that are eating you. So there seem to be some underlying laws that determine the abundance of all these things. I'm not going to talk much about that. Okay, so essentially, the bottom line of that is there are undiscovered viruses everywhere. Dressing up as a tropical virus hunter is fun, but you might find as many in your own backyard. To understand which ones of these are going to be important, we need to do something about their population dynamics. And tragically, just finding them and looking at their DNA doesn't tell us anything about their population dynamics. The question I always ask myself, would the virus hunters have detected HIV? If you'd brought back the RNA, looked at it, done some initial tests, you would have said, this is a relatively innocuous virus. It's been in these people for 3 or 4 years. It's not doing anything. You would have completely missed it. And indeed, the key thing, I think, for many of these things, nothing we can get from their RNA or their DNA tells us 2 vital things. What is their basic reproductive rate? How quickly does the first infected patient infect other patients? And what is the proportion of time that the pathogen is infectious before symptoms appear? And just on here, we can see that SARS has a relatively low R0, and symptoms appear almost as soon as you're infectious. Similarly, smallpox. Influenza has a much higher R0, and symptoms don't appear until you've been transmitting it for 2 or 3 days. The really scary thing about HIV, R0 is about 4 or 5, but you can be infected for 8 to 9 years before you ever show symptoms. And those are the ones we should be really worried about, because in terms of controlling them, our ability to control them depends on how far they are to the left of these lines. It's really easy to control something like SARS. It was relatively easy, even though it's a huge medical thing to get rid of smallpox. But influenza and HIV are much, much harder because of this long period when they're transmitting before you know you're sick, and because of their high R0s. And the DNA and the RNA tells you nothing about that, because we can't do the predicting up from the DNA of the pathogen down to the DNA of the host. We need to actually get experimental data on that. Now, let's go to one of my favorite places, the Serengeti, for the next vignette. Again, this is a similar diagram to I showed you before. This is a food web for the Serengeti with all the species you see on TV in red, the sort of plants and the wildebeest and the zebra. The yellow dots are just the parasites from the wildebeest and the viral pathogens from the lions and the domestic dogs. If we could put together a complete pathogen map for the Serengeti, there would be as many or more free-living species than there are, parasitic species than there are free-living species. By we I mean sort of Sarah Cleveland, Craig Packer, and a wonderful Katie Hampson, Tiziana Lembo, have been looking at parasites, viral pathogens of carnivores. These appeared as a problem in Craig's Lyme project when distemper spread from domestic dogs through the Serengeti, causing a big die-off in the lion population, perhaps interacting with another pathogen, as I'll talk about. So what Sarah and the rest of us decided to do was to vaccinate domestic dogs against 3 different viral pathogens. Rabies, because it was a big problem also for human populations, but also canine distemper and canine parvovirus, 2 viruses. We were very successful in reducing rabies, because it had the lowest R naught, but we started looking more detail at distemper and parvovirus, and we produced a paper on this last year, but Sarah said, it's really weird, we're vaccinating like mad, and it doesn't seem that we're having any effect. And if you can see, the vaccination coverage has been going up, but the actual level of dogs seropositive in villages where we've been vaccinated hasn't really changed that much, partly because they're getting converted by the vaccination. Now, what's happening here? What happens if we move from our usual comfort zone of looking at models for 1 pathogen and 1 host and start looking at models for 2 pathogens and 1 host, little stumbles before we start running? If we do that, it's relatively easy to make a model for canine distemper, and it's really boring. It does everything you would expect an SIV, an susceptible infected recovered model to do. You introduce the virus and it settles down to steady dynamics with lots of hosts that survived, recovered, relatively constant abundance of each. If you plot it out, susceptible hosts against infected hosts, you get this nice smooth circles. If we do the same thing for parvovirus, it's even more boring because it's got free living stages that really help stabilize everything. So each pathogen by itself has really boring dynamics. What if we put both of them in at the same time? When we do that, the dynamics begin to get more interesting because you can be infected first with 1 pathogen and then the other, or first with the other pathogen and then the first one. But even though no 2 hosts are ever co-infected, the dynamics become richer and more complicated. And indeed, when you plot them out as susceptibles against infected, you can see that many more complicated things are going on just by having 2 pathogens in the same population. And that's something you wouldn't get if you just made your model about the 1 disease you're thinking of, or just tried to interpret the data on the 1 pathogen you're interested in, or both of them. Until you've made a mathematical model, you don't get that understanding of what the dynamics are likely to do. Instantly, one effect of that is having 2 pathogens increases the persistence time of either of them in the population. And that's an important effect because although we spend a lot of time focusing on how the pathogens establish, our next big worry is how do they persist, and having 2 pathogens in a stochastic population increases their persistence over having just 1 of them by themselves. Secondly, how does it affect the way they respond to vaccination, a control measure? And we know we have really good vaccines for both distemper and parvovirus, but you can see here, what I've plotted out is the proportion vaccinated against the numbers infected with just red is canine distemper virus, blue is canine parvovirus, and then the little dots are it had parvo, it had distemper, and now it's got parvo, it had parvo, and now it's got distemper. As you vaccinate, you have no effect on that. Why do you have no effect? When you look at the top line, what you're doing is you're just producing more dogs. As you vaccinate, dogs live longer. If they live longer, they have more puppies. So you end up with more and more individuals that have survived and recovered, and you have no change in the abundance of susceptibles because the disease keeps going by holding the host population at a relatively constant level of susceptibles in the population. And it's not until you've got to the level of eradicating one of the diseases, and the parvovirus goes first, that gives you even more puppies being produced, and then you need to get up to vaccination levels of about 90% before you get rid of them, because the more they vaccinate, the more puppies you produce. And that's a different calculation than the one we do for vaccinating in human populations, because despite our own personal excitement, the birth rate of human populations is really slow compared to dogs. That means you can also substitute at certain levels sterilizing dogs for vaccinating them, and that allows you to get rid of the diseases much more quickly, except people in Tanzania do not want us to sterilize their dogs. But again, you wouldn't see that effect, and you couldn't predict it unless you actually did the modeling. Do we see any evidence for that? Well, a wonderful paper that came out just before Christmas is by our wonderful vet, Anna Chapina, working out of Lincoln Park Zoo in Chicago. She's been comparing villages where they've been vaccinating with villages where they haven't been vaccinating. In the vaccinated villages, initially, you see the dog population increasing much more rapidly than in the non-vaccinated village, but eventually the rate of increase slows down. Why does it slow down? Well, it turns out if you look at the mortality causes, what you're doing in the vaccinated villages is having many more dogs being eaten by hyenas. Essentially producing all those puppies by vaccinating the dogs just produces more food for the hyenas to eat. So instead of dying of sickness, they're dying by being eaten by hyenas. Which again tells you thank you from the hyenas. To understand how these things work, you need to embed the dynamics within the larger food web. Now let's go to Brazil. Often a fun place, but not a fun place last year was Zika virus. This was a pathogen, as I said, it was first described back in 1963. The total number of world cases was trotting along. It suddenly took off in Brazil. It's taking off in Colombia now. It seems to also be in Indonesia. But it has this very dramatic dynamics of going up and then coming down again. Now, because the Olympics were due to be held in Brazil last year, there was a huge panic of what you do with mass public spraying to try and kill the vectors, which themselves are an invasive species, the Aedes aegypti mosquito. We, like almost every other breathing epidemiologist on the planet, thought we should model this. This is going to be really exciting. And there's the usual frenzy of a race to get your paper to Science first. But we thought we have a start on everybody because we have a Gates grant to look at Chagas disease, which is a vector-borne disease throughout Central and South America, killing about 5 million people a year. So using this sort of age-structured vector-borne disease model, we did the sort of classic Keith Richard, Mick Jagger thing. We've got an interesting song here. Let's speed it up and turn Chagas disease into Zika by making the dynamics much faster, which is relatively straightforward. We knew this model works pretty well for Chagas because we can make up to 10-year predictions for numbers of Chagas cases with different age classes. So we just speeded it up, converted people who are acutely infected into resistant hosts, and began to say, well, what would this do for Zika? And the results are really quite interesting. This is looking at the number of Zika cases if you did nothing. So this is no intervention. The red line here goes up. You get about a peak of 4,000 cases in the first sort of 3 or 4 months of the epidemic, and then it dies down again. It dies down because essentially it's transmitted so efficiently that everybody becomes immune. If you kill 80% of the mosquitoes, you slow the epidemic down, but you don't change the number of cases. The epidemic just goes on for longer. If you kill 90% of the mosquitoes, you slow the epidemic down hugely. It lasts nearly a year, but you don't reduce the number of cases. It just takes longer for it to go on. And that's because in these vector-borne diseases that produce complete immunity, the dynamics is driven by the buildup of herd immunity. Once the mosquitoes are biting, if you slow them down, you're just prolonging the epidemic. And that's not a good result for public health intervention. You have to be seen to be doing something, but one of the worst things you can do is inefficiently spray to kill the mosquitoes because you're just making the thing last longer, and that would allow it to go to other locations. And that was a result that didn't appear at all in any of the literature on Zika. We sent it straight off, but of course we were beaten by Neil Ferguson, who had been to Brazil and got lots of the data. He was essentially showing the same thing. If you read what Neil's paper in Science said quite carefully, the intervention doesn't do a lot of good because the herd immunity that the disease generates will shut the epidemic off, and you won't see it again for about another 8 years, when it'll come back as a much smaller epidemic, 1 or 2 orders of magnitude smaller, just reflecting the number of people who've been born since the previous epidemic. And that, again, is a chunk of theory of what happens when these vector-borne diseases establish in host populations. The dynamics are entirely dominated by herd immunity rather than what the vectors are doing. And that's a very worrying result. You can formulate that into the standard way we tend to look at vaccination as a control strategy. This is just saying, what's the R-naught of different vector-borne diseases, Zika, dengue, and malaria, against the proportion of mosquitoes you have to control, the blue line, versus if you had a vaccine, the proportion of you'd have to vaccinate. And vaccination is so much more efficient than mosquito eradication, partly because vaccination gets you double the money. You protect the hosts that do get vaccinated, but you also protect the mosquitoes from getting infected when they bite the people who were vaccinated. So vaccination is much, much more effective against vector-borne diseases than mosquito control. All of the NSF money went to mosquito control and understanding mosquito dynamics. Notice also another important effect. If you plot R0 against average age of infection, what you really wanted to do with Zika was get the average age of infection below child-rearing age. The disease does that itself really, really efficiently. If you kill the mosquitoes inefficiently, you keep the average age of infection up and increase the number of women who are likely to have babies that aren't fully functional. So again, that's a worrying result that you wouldn't get if you didn't do the mathematics, and you wouldn't get that from virus hunting and finding it. Okay, so that's 3 vignettes of some of the things we need to be doing. For the major chunk of the talk, for about the next 20 minutes, 25 minutes, I'm going to talk about this scary creature. This is one of my favorite birds. It's a house finch. It's the host to an emerging disease. Because house finches are themselves an introduced species, nobody really cares about them apart from some fanatical ornithologists. So we don't intervene at all with this disease. It's work that's been done, again, sponsored by the Ecology of Infectious Disease Project, NSF, based in Cornell, but with a large group of people who are now also at Emory, University of Wisconsin, Madison, University of Connecticut, a whole bunch of different places. If we go and look at the finches, their natural habitat is actually the deserts of the Southwest. They're happily living in the Sonoran Desert, which is their natural habitat. But they were introduced by one of these bird fanatics into Long Island in about 1940, where about 10 house finches were introduced and essentially did nothing for about 10, 20 years, until about 1960, 61, when their numbers began to increase. And this is the movie part of the talk. This is the numbers of house finches counted coming into people's backyard feeders by the Lab of Ornithology at Cornell's backyard feeding census. So through time, you can see the house finch population beginning to expand and boom. Around about the 80s, it really begins to take off and spread west to join the native populations in the west of the country. In about 94, something interesting happens. A disease emerges, and notice the numbers start decreasing in the east. The size of the circles gets bigger, there are more holes in it, and the abundance of the finches declines dramatically in the east. What happened then is a bacterial pathogen, Mycoplasma galliseptum, that's very common in poultry, jumped into finches. And it probably came in from backyard poultry farms. You could have a little backyard herd of poultry and just for your own personal use, and it can be up to 25,000 birds, which is an interesting number of pets to have, but you don't get above that until you don't become a commercial enterprise, so you get above that and put them in a shed, which means that they can interact quite happily with these house finches. And what is usually a gut pathogen, an enteric pathogen in these chickens, becomes an eye infection in these finches. And the nice thing is that when it's not really nice, it's not nice for the finches, but the people who look at birds coming into their backyard bird feeders can identify birds that are infected with this. So if we go back to, this is just the boom in house finches, 1993, we know that this jump across occurred around October 1993 and began to impact the house finches. So suddenly we have an emerging disease in a host that's common throughout the US, and we have many volunteers looking at birds at the bird feeders to see if they're infected or not. So we have an epidemic of an emerging disease we can actually monitor to see how it spreads, and then by sampling the birds, collecting the bacteria, doing experiments on them in the lab, we can look at if this pathogen is evolving or not, and how its interaction with the finch changes through time. Now, as I say, it's intriguing because one of the things this pathogen does, it usually lives in the guts. It now lives in the eyes, which causes the eyes to flare up. So it's easy for people to identify if a finch is infected or not. This caused Andre Dant, as soon as it occurred, to add an additional feature to the usual backyard feeder surveys that the Lab of Ornithology does, we started doing a house finch disease survey in which people were asked if they could make sure they looked at their bird feeders for 25 out of 30 days every month and record whether they were seeing healthy or sick house finches, goldfinches, house sparrows, and any other infected birds. So it's a wonderful, and these are the sort of volunteers. There are 25,000 people who are happy to spend a couple of hours each day looking at finches coming into their feeders. Plainly, the distribution is not random. There are many more people closer to Ithaca than there are as you go further away, but we've got pretty good coverage across the entire eastern US and across to the west as well. What that showed was the sort of laminar spread of this pathogen following the advancing front of the finch population and spreading just behind, almost overtaking, in fact, eventually overtaking the spread of the eastern birds as they spread towards the west. Now, we could do, with David Lee, we did quite a lot of poking birds in the eye with sticks, but this is all done in ways that do not offend the animal care committee. This is a Q-tip. You put it in the corner of the eye, you squish it around a bit, and then you can grow it up to show that there is mycoplasma growing in this eye. You can then take the Q-tip, another Q-tip, stick it in the eye of an uninfected bird and show that that MG, what you've transferred, will cause the same infection, the same symptoms in the other bird. So we can do all the postulate stuff. This is the finch. 1 hour after you poked it in the eye, it's fine. They don't mind being poked in the eyes because you don't actually puncture the skin. What they do happen is this is what happens at the population level. These are finches in Virginia, Maryland, and Delaware. These are ones in central Pennsylvania and New York. This is the population initially increasing. This is when the disease comes out. It knocks the population down by about 60% in both places. And that's of the order of 500 million finches when you add it up. You know, there were many happy house cats around, and raccoons, I suspect. The way that interacts is quite interesting because it doesn't have an impact on the geographical range of the finches. The finches continue to spread. The biggest effect is on the social group size. The finches used to live in groups of the order of 12 to 16. You never see a group of finches bigger than 6 coming into your feeders now. It completely changed the size of the social group in which the finches lived throughout the East Coast. We could then also begin to collect data and plot the spread, and also by doing infections on the birds and looking at how quickly their eyes get infected and how badly they get infected, we could look at how virulence changes. So if you stay on the East Coast at different locations where we're sampling them, these are different sort of genetic trees that begin to establish locally, it gets steadily more virulent at all locations on the East Coast, and then it suddenly spreads west, makes its way to California, and like all the rest of us, it becomes much more passive, much less aggressive, and becomes almost avirulent on its way to California. Once it's been in California for a while, however, it flips around and gets much more virulent and unpleasant again. You know, probably went to LA or San Francisco or something. So we can both plot out how it's spread through time and how its virulence has changed through time. And again, everybody's big worry was about Ebola and other things. They're all going to evolve to get more virulent. This did both. It evolved to get more virulent in the same place. It evolved to get less virulent as it spread across the US. Now, what would cause that? We know the house finches were an introduced species on the East Coast. We know they're native on the West Coast. They live in much smaller social groups in the West. So it may be that just transmission is less efficient in smaller social groups, so it has to be less nasty in the West because the social groups are smaller. It could also be, this was interesting, this was all of a beautiful fuzzy slide before, which often happens to me when I look at genetic slides, but this one is actually looking at the allelic diversity of house finches. In the West, they're much more genetically diverse than they are in the East. So maybe the difference in virulence are due to the genetic background of the host. So to look at that, we took the parasite strain that had got to California. We keep the strains going in the lab, the parasite strain that's in the East Coast, and take a bunch of birds from the West Coast, a bunch of birds from the East Coast, set up an experiment where we just infect the birds and look at the difference between them. These are the initial results, or essentially looking at how unpleasant the eye score is through time, which you can divide up in 2 ways. What is the difference between the hosts on the left And there's no discernible difference between the hosts. The genetic background of the host is having no effect on the pathogen. The pathogen is consistent, whether it's in a genetically inbred or an outbred bird. It's dependent on whether it's a nasty pathogen from the East Coast or a mild one from the West Coast. And that's really quite funky. It says the host genetic background has no effect. Now, we can do a bunch of things. I've seen I'm sort of beginning to run out of time. One thing that's happening that we consistently see is this parasite produces immunity, and through time, as it evolves, that immunity becomes asymmetrical. I'll skip over this, but essentially, once it got to California, it got more and more aggressive, as I said. So as it goes up to match the levels of aggression, of virulence that we saw in the East Coast when it first emerged, having initially become more passive. We know it's only jumped across once. All of the strains we see come from this initial crossing over event. What's quite interesting is we did pick up, there's a fantastic tree for this mycoplasma because it's in many domestic poultry herds. A different strain did try to jump across, and we picked it up in a house finch in upstate New York, but it died out. So all of the strains we're actually seeing come from one initial crossing over event. They are split now into two subpopulations, one for the East Coast and one for California. The East Coast one, as I said, evolved to get more virulent. The California one initially got less virulent and then more virulent. We can make models of this. We can essentially, I'll skip over this, but essentially putting together a model that looks at trade-offs between birds being able to disperse and the evolution of virulence shows you that if you have no trade-off between movement and virulence, the pathogen will evolve to get more virulent. If you do have a trade-off, it will evolve to get less virulent until it doesn't have to move anymore when it flips and starts being more virulent again. And we can show that that's all driven between trade-offs between pathology and transmission. And indeed, what seems to happen, as it evolves, it gets more virulent, and the more recent, more virulent strains can exclude the earlier strains from circulating in the population. So as the pathogen becomes more diverse, more virulent, it's able to exclude earlier strains from being able to reestablish. So we can do that comparing across strains and show that the later and later strains are both more virulent and better at excluding the earlier strains. Now let's quickly skip to, we could do the stuff that shows that the more virulent invader will exclude a less virulent one. What is the mechanism of that? So just to wind up, the mechanism is really interesting because working with groups of people both at Harvard and at the University of Connecticut, we can show that the genetic changes in the bacteria are all occurring at the CRISPR locus. Now, if you say CRISPR at a microbiology conference, it's a bit like going to one of those Beatle concerts in 1960s. So we're not going to do any fancy genetic modification with CRISPR, but why is all the selection occurring at CRISPR? Well, what seems to be happening is, let's think what's going on. Mycoplasma has a CRISPR locus, and that CRISPR locus, or set of loci, is set to fight phage. Now, if you're living in the gut of a chicken, it's a really disgusting place full of unpleasant things. A bit like, a cabinet meeting of the new Washington people. The good thing of all those disgusting things, there are things all trying to kill each other, particularly phage trying to kill the bacteria. Once you're in a nice, healthy immunoprivileged site, like the eye of a finch, there is no phage there trying to attack you. So you just don't really need your CRISPR locus, which is essentially a bacteria's equivalent of its immune system. So that begins to atrophy and become things that increase your ability to attach. So the increased attachment allows you to grow more aggressively, become more virulent. You don't need CRISPR anymore. So the miraculous thing that seems to have happened is that this pathogen has become more virulent by losing its own immune system equivalent to turn it into something that makes it attach more readily and perhaps reproduce more readily. And I find that really funky, that this pathogen is actually escaping its own pathogens, is its way it's evolved to become more virulent. Again, something you wouldn't pick up if you just went the virus hunting. So what have I tried to say? So I say the emergence of the mycoplasma in house finches provides, I think, a supermodel system for looking at emerging diseases and provides important lessons for the other emerging diseases that we keep trying to stop before they move over and kill us all. The nice thing about the house finch project is we've been able to look at it as real systems biology. We go from genomics of the phage of the bacteria through the genomics of the bacteria up to what are the continental level impacts of that pathogen on finch and other bird communities. So that's what I call systems biology, not just, oh, we looked at this gene and we think it has something to do with the liver. The bottom line, should we be worried about emerging diseases? I think yes. If you believe in God will save you and you don't believe in evolution, you should be very worried about emerging diseases. If you believe maps will save you and we can make vaccines overnight, you should be very worried. Two things don't cause me to be worried, and give me optimism. This whole field that started with this Ecology of Infectious Disease program has produced some amazingly bright young people. And they're onto this problem. Witness the competition to get these papers out on SARS every time a new disease appears. We've got some super bright people out there doing things, and that's a good thing. And if we could change the playing field in the way this was all funded, we would actually also do much better. Again, the bigger pictures, ecology and mathematical biology can supply a significant number of insights into this, as much as just doing the genetics and the virus hunting. The population dynamics that go back, Anderson and May, Kermack, McKendrick, Ross, that is the macroscope that's the equivalent of the pathologist microscope. We won't understand diseases unless we use those tools. They're the most powerful tools we have. And that should also be central to this whole new One Health, eco-health agenda. I'd like to acknowledge many people whose names are up here. Everything that I've said more or less came out of conversations with them. Okay, thank you very much.
Thank you, Andrew, for a very provocative presentation. Now we're fortunate to have as our faculty commentator Jessica Hellmann. Dr. Hellmann is the director of the Institute on the Environment, as I mentioned before, here at the University of Minnesota. She is also the Russell M. and Elizabeth M. Bennett Chair in Excellence in the Department of Ecology, Evolution, and Behavior. Professor Hellmann's research focuses on global climate change, ecology, and climate adaptation. She has led an important paradigm shift in ecology and natural resource management by showing that adaptation, living with climate change, is just as crucial as to the future of humanity and the Earth's ecosystems as slowing and stopping greenhouse gas emissions. We're very happy to have you here today to comment on that provocative presentation. Dr. Hellmann, thank you.
So the fun thing about being the respondent is I'm the one who's keeping you all from asking Professor Dobson the questions that you have in response to his presentation, at least slowing us down. Sorry, what did you tell me? Forward, backward. Got it. I can do forward and backward. Okay. Well, let me start by saying thank you to Professor Dobson for thought-provoking remarks. You help us see the interconnectedness between ecological systems and human health dynamics. And I think you really showed us the power of reframing, that if we think about disease in different ways, if we think about reducing and influencing the dynamics of disease, instead of always just imagining eliminating or curing them, we may actually stand a better chance of improving health for everyone. Thank you also to the Consortium on Law and Values in Health, Environment, and Life Sciences for the opportunity to be here for this event, and for the opportunity to add just a few remarks on the subject of Professor Dobson's lecture. So I get to have a moment to add another layer of richness and context to Dr. Dobson's presentations. And I'm going to speak just very briefly about the influence of, shockingly from my biography, climate change on disease, including emerging infectious disease. So my goal here is to bring up another feature or dimension of today's conversation and another aspect that strongly affects disease dynamics and how we manage disease. And this is in no way to distract or contradict many of the important points, Andy, that you raised, but an attempt to enliven the conversation still further. So the effects of climate change are beginning to appear everywhere around the world in the form of steadily increasing global temperatures and year after year of record-setting weather events. And this climate change will accelerate as we approach the middle of this century. By the end of the century, if we follow a business-as-usual scenario, between 5 and 9 degrees Fahrenheit, 3 to 5, 6 degrees Celsius, warmer than our historic norm. By the after 2100, by the end of this century, as we reach a new equilibrium of global average temperature, that could be between 5 and 20 degrees Fahrenheit warmer than our historic global average. That happens long after, several hundred years after we stop releasing CO2, methane, and other greenhouse gases, as we reach this new stable equilibrium. And we will stop adding greenhouse gases even under a business as usual scenario because of technological innovation and decreasing availability of fossil fuels. If we are more aggressive in transitioning our economy to renewable and carbon neutral energy sources, however, we might be able to reduce the amount of climate change to a lower level. And climate and weather, we know, affect human health directly in the form of natural disasters, crop productivity, heat-related deaths, aggravating air pollutants, and other such forces. But a changing climate also influences health indirectly by affecting the pathogens in the community of pathogens and vectors that make us sick. So, for example, climate modelers and public health experts are predicting increased risk of disease, or increased risk of food, water, and vector-borne diseases, with some evidence of that change happening already and more predicted as we accelerate warming. We know moderate increases in temperature, for example, and precipitation affect positively the vector of dengue, for example. However, as Dr. Dobson suggested in his remarks, there's quite a lot of uncertainty, a lot of complicated interactions in what drives actual disease outbreak and disease dynamics. Furthermore, humans have a lot of interference, hence we need to go study birds instead because the mark of human intervention in disease risk and spread. We use bed nets, we use vaccines, we have wastewater treatment. If we add to that those complicated dynamics and that signal of interference, we add uncertainty about how much greenhouse gases we will add to the atmosphere, and we add uncertainty about how climate change will manifest in particular locations. We don't yet have good models that tell us very fine resolution about what the consequences of climate change will be. This makes it very hard to make specific predictions about rates of disease and where new disease will emerge. I'll come back to this. But we do know with certainty that climate will change, is changing, and this will make new people sick in new places. And we know that this changing climate invites us to reevaluate and perhaps adjust our disease protection strategy. Which brings me to this photograph. Recognizing that climate change affects all aspects of human and non-human life, our global society has been steadily working toward agreements to slow and then eventually stop greenhouse gas emissions. And the latest international agreement in this process, the culmination of 21 years of negotiation, was reached in Paris in late 2015. Now, the most well-publicized part of the Paris Agreement is the bit that commits signatories or countries around the world, including the United States, to greenhouse gas emission reductions that keep the amount of global warming to 2 degrees Fahrenheit, or 2 degrees Celsius, or about 3.5 degrees Fahrenheit, above our historic average. We furthermore say maybe we'll be lucky and we can make that 1.5 degrees C. And at present, countries are working toward this goal with voluntary commitments to greenhouse gas reductions, and those commitments manifest themselves by 2025 and 2030. And then greater commitments and reductions are made thereafter. So under the Obama administration, we were working toward a voluntary commitment of reducing emissions 25% below 2005 levels by 2025. And we were doing that via the Clean Power Plan, which you might have heard is a plan has already been facing federal litigation and is slated for the chopping block by the Trump administration. Now, when we look at the Paris Agreement in its entirety, we see this word mitigation to stop the emissions of greenhouse gases 50 times. It's a big focus of the agreement text. But less well known is the prominence of the word adaptation in the Paris Agreement, which you actually find referenced 85 times. Adaptation is all those things that we will do, that we will be forced to do to live with climate change. The ways we have to adjust our lives, the way we manage natural resources, and the amount and the way that we combat infectious disease and other threats to human health. And we will do it regardless of which presidential administration is in power. But it's going to require considerable time and resources. And how much we'll have to adapt, of course, depends on how much we allow the climate to change. Now, there's only one part of the Paris text that is quite specific about adaptation, despite the word being referenced lots of times, and that's a phrase about raising money for both adaptation and mitigation activities, encouraging voluntary commitments by wealthy countries to a fund reaching $100 billion annually by 2020. We are nowhere near that target now. In fact, only about $10 billion have been committed so far. But the magnitude of this goal reminds us how large and daunting adaptation could be. And it showed the fundraising goal shows us the relative lack of resources that many developing countries have relative to developed ones. So much of the adaptation that we're going to need for human disease is straightforward. Climate change is likely to exacerbate some disease risk, and we're going to confront this with traditional means. Improvements in basic health measures such as sanitation, essential healthcare services, disaster response, poverty alleviation, for example. Many of these focus on the herd, not the vector, as Professor Dobson discussed. But, and this is where I think maps matter, and we can debate this later here, for those basic strategies, including managing herd immunity, to be effective in the face of climate change, we have to direct them to places where we imagine new disease outbreaks are most likely. Not necessarily emerging new diseases, but even some of the existing diseases that we have appearing in new locations. We have to anticipate changes in the effectiveness of vectors and of our control measures under different climate conditions and identify places where disease was once held at bay, but because conditions are changing, new infections can arise. To do this, however, we will need the strategy informed by the techniques that Professor Dobson outlined in his presentation, including mathematical models and experimental research that reveals key mechanisms. Like most ecological systems, the effects of climate change are not going to be so simple as just to cause an increase or a decrease in disease. It's going to perturb an integrated system, much like the food web diagrams that Professor Dobson showed, where diseases are incubated and transmitted. And process-based mechanistic modeling can help us understand those connections. However, we have yet to pursue this, let alone complete it with a climate change perspective. So if there is an upside, or the upside if there is one, to addressing climate change and adapting to it, it is that it actually invites us, I think, to be smarter, to be more efficient, and to be more scientific in our global health work than ever. Those young people that Professor Dobson referenced growing out of, for example, the disease ecology program funded by NSF are going to be busier than ever. Still, a prudent society would stop and slow and stop factors like climate change because they will cause a shift in that complex ecology of pathogens to lead to new infections in new people and places. But with climate change upon us and society still crippled with debate about which scientific facts deserve our respect and attention, we have little choice but to come to terms with a world where climate molds and modifies our every being. So in my brief remarks, I invite you, I'm asking you to, in addition to Professor Dobson's remarks, to consider the intersection of what you heard in his analysis with another emerging major force of change in the form of climate change. Thanks very much.
Thank you. Okay, we have time now for questions. Please, again, use one of the two microphones. If you're going to leave early, please go out the side doors here so you don't block the cameras. Don't walk across here. We have this on webcam right now, and it'll be also later on tape. So anyone with a question, please. Well, I'm going to start with the first of all. You hinted in here that the virus hunters were not necessarily at the top of your priority list for dealing with or responding to the issue of emerging infections. Could you elaborate on that more and what your thoughts are on that?
I think the virus hunting thing has been oversold as a way to go. It is the current largest part of any money being spent to look at emergencies is being spent on people just to make longer and longer lists of what viruses are out there, searching for new ones. Because I think it's valuable to build up that database, particularly the genetic database, so when something new appears, we can quickly identify it and where it's come from. But I think to get a better understanding of how we're going to deal with the next SARS or the next Zika, we need to invest more in training people to do the mathematical modeling and getting a deeper natural history understanding of how different types of viruses work and why we get these subtle but really important differences between something that's really benign in a bat that suddenly becomes incredibly pathogenic in a human. Or why do the viruses or the bacteria express themselves so differently in different host species? And we're not putting enough energy into doing that, or resources.
Okay. Again, questions in the audience here? One over here.
I also think that the maps themselves are very misleading. We've had fantastic developments in geographical information systems. Those are wonderful, but they can make, as I said, 200 data points. Doesn't give you something you can make a global prediction with. It's misleading would be the gentlest way to describe it.
But Andy, could you say, what are the maps attempting to show?
The maps are attempting to show where are the hotspots where we would expect new viral diseases to appear from.
Based on where previous ones have emerged.
Yeah.
Well, if I could just add before I ask questions, since we're challenging the norm here, I think that one of the biggest problems we have today is we have an epidemic of modeling going on. And saying that to a modeler, I brought front and center, because while some models surely may inform us, we've been misled so many times by modeling that you've really set a tone very differently. When the CDC modeled that up to 1.5 million cases of Ebola might occur in West Africa, or if you look at any number of different disease issues, HIV in the early days, we look at bovine spongiform encephalopathy in England in the early days, the models typically were so far off that they caused us great challenges where we had to go back and try to gain credibility. So while modeling, I think, can be also very helpful, I would say in the maps, I would come back and add that we have to be very careful about our modeling today, and while it can be very informational, it also can be very, very difficult.
Well, it depends on who you get to do your modeling.
Those maps aren't models.
See, when you do it, I know it's right.
This is going to get exciting.
When I do it, I'm always slightly cautious. I think the vital thing with the modeling is as with anything else, is to state what your assumptions are rather than what the results were. And that CDC prediction for Ebola, instantly other modelers said, that can't be correct. Right. Your assumptions are deeply flawed. There was a very healthy discussion with the BSE epidemic between the top groups of modelers, and they got really much more correct than they got wrong. And it was a healthy discussion that sort of said that we all agree on that assumption. This is something we don't know about. We could go to one or two things based on what we do know, but we desperately need more information about it. So the power of the models isn't just what they tell you. It's when you hold it up against the data and say, it fits here and it doesn't fit here, so we need to know more about that. So it gives you confidence in what the assumptions being correct, but it also tells you what you need to know next.
We could go on with that one. I can just tell you, I've never been at a congressional hearing yet where I was at the witness table getting grilled by congressmen, why the hell did public health so exaggerate this? None of the models were ever at the table. It was always us trying to explain for them. So we need to do more on that. So, yes.
I'm happy to go and talk to it.
Thank you. Thank you.
Start with talking about the models for.
Please, go ahead.
I'm wondering about the generality of the defense attachment trade-off that you saw on the Finches, or what are the mechanistic basis of that trade-off is understood?
No, I mean, that's a very interesting question. We're just beginning to explore in more details what's happening with how the bacteria evolves. The defense mechanisms, there's a whole bunch of stuff I didn't talk about. Again, I had some stuff on climate that I actually took out, which I should have left in. But what we've been doing is when we poke the finches in the eye with a stick, we can actually have a more subtle stick, which is a calibrated piece of filter paper. So we can both, by just gently touching the eye for 15 seconds, we can quantify the bacteria abundance in the eyes, and we can quantify the abundance of the two immunoglobulins that are attacking the bacteria. So we can do experiments where we can infect one eye and then monitor the bacteria in the infected eye and the immunoglobulin response in the infected and the uninfected eye. So we can actually look at the dynamics of the interaction between the immunoglobulins and the bacteria to see how they're fighting with each other and what's the dynamics of that, and how does that in turn lead to the production of the secondary things that lead to the inflammation. So again, it's an intriguing pathogen in that lots of the transmission goes on in that rapidly reproducing phase of the bacteria before the immunoglobulins catch up with it. Once you begin to see the pathology, the transmission's almost stopped. So in some ways it's flipped around from those other things where you see the incubation is when lots of the pathology is going on, and then you don't get the transmission till later. Here, lots of the transmission is going on early on. So we're beginning to be able to do the within-host modeling of that. It would be fascinating to do the same thing, going to look at mycoplasma in the guts to see what is the interaction between the different parts of the immune system attacking it in chicken guts, and how is that interacting with the phage that's also interacting the bacteria, and the things operating in different ways. And indeed, we've also tried to do that in other, we know we can put mycoplasma through some chickadees, and we can put it through house sparrows, and we can put it through juncos, but we don't get the massive inflammation. So we're trying to sort of get a finger on how does the within-host dynamics change, and is that because of different immunoglobulins or different rates of production of different immunoglobulins and attacking them? But again, I didn't talk about that, but it's something I'd be happy to talk offline about.
Good. Any other questions? I have a set of questions that go back and forth. So what do you think the most important take-home message from Andy's presentation is for climate change? And in turn, from your work, what do you think the most important implication is of climate change, and as we've heard from Jessica, adaptation as it relates to yours? So let me ask you first. What was the most important lesson for you in climate change?
Well, I'll follow then. I guess my answer would follow on your critique of models in defense of them. I do, for full disclosure, I'm an ecologist, so my view of, trained to think of disease dynamics like dynamics of other living things. But I think the opportunity to ask a model of the kind, for example, that Andy showed with Zika, invites you to step back and say, what is your desired outcome? What is it that you're trying to prevent from happening? And I think when you imagine an additional stressor like climate change coming through the system, again, it asks you to say, what is the public health outcome that you are trying to achieve? You may, and with a modeling framework that includes some mapping, you may identify new geographic areas in which you need to apply these modeling techniques. But the model allows you to build a toy system and say, what do we think the strategies are that might be effective? So to that point, I think many of these mechanistic models are less about how many people are going to die of Zika or of Ebola, but rather what are the tools that are available to us on the table, and what are our relative confidence in the different tools in being effective for our particular outcome. And I think all that climate change does in a public health context is layer on top of that and invite you to think about new geographies and new dynamics, and taking those kinds of food webs that Andy showed in his talk and moving them around, squishing them and pushing them and prodding them in different ways. And the only way you would ever make sense of that is by stepping back and trying to make some mathematical or pictorial representation of the dynamics that are going on in that system.
Yeah. Which point taken? Yes. Andy.
Well, I didn't talk about climate change. I had a sort of 15-minute things about the work we're doing up in the Arctic and why we're working up in the Arctic, but I didn't put that in. The reason we're doing that is, we're all very worried about climate change. The place we have the strongest climate change signature is the Arctic. But we also have relatively simple ecological systems up there. So we wanted to go up and work up there to look at systems where we had 1 or 2 host species, 3 or 4 parasite species, an incredibly strong annual climate signal that's also changing very rapidly. The one thing that we see there that's a very, very strong effect, that our initial prediction with climate change is that the heights of transmission that you get at the peak seasonal time are going to get bigger. We thought we could easily show that. It turns out that that's wrong. What actually happens is it gets so hot in the Arctic that the processes slow down, and instead of having 1 peak, you get 2 peaks, 1 in the spring and 1 in the fall. So the transmission seasons get much longer, and instead of hitting a non-linear system with 1 kick a year, you hit it with 2. And that's going to give you much more complicated dynamics. And so straight away, we thought, we wouldn't have understood that if we hadn't done the models and started looking at what climate's going to do, just based on thermodynamic principles in these systems. So again, and that's also something you wouldn't, there's a huge debate about the importance of climate and malaria and climate and cholera, and that's because those are tropical diseases. The biggest things going on in the tropics is land use change. And it's hard to disentangle climate change from land use change. And that will always cause debates that can become divisive and give the people who don't believe in climate change ammunition to say the scientists don't know what they're talking about.
Thank you. Susan?
I wonder if all 3 of you could kind of dig in to this category of emerging disease to differentiate scale, epidemics, pandemics. I mean, what do we know about our vulnerability, whether the dynamics are very different depending on which of those categories you're talking about? And for Jessica, whether climate change has particular potency depending on what scale of emerging disease you're talking about. So inviting you not just to put everything in the bin of emerging disease, but to sort of disaggregate.
Well, let me, as moderator, in a sense, just try to take a crack at that, and maybe you. First of all, I really want to congratulate Andy. I thought that the discussion of looking at infectivity, reproductive rate, and incubation period, and when one becomes infectious is all very critical. I mean, to me, it's kind of like when the pitch is thrown at the home plate, how does that center fielder know at the sound of the bat ball hitting it where to run? He doesn't sit there and think about it. It's a whole series of things that happens, whether it's his eyes, his ears, his experience. And as an epidemiologist, we take all the things that Andy just said into account all at once and say, oh my God, this is going to be a bad one, or well, that's not so bad, we can handle that. And I think that today, clearly, anything that's a respiratory transmitted pathogen, that you are infectious prior to the onset of symptoms, and also for which there may be a relatively high pathogenicity, ability to cause disease, high virulence, i.e. ability to cause death, is one of great concern. There is still no other disease on the face of the earth that will equal influenza. It is the Lion King of infectious diseases. It has been. I really believe that it will continue that way. I can't imagine another pathogen, HIV, anyone could combine. We sit today, Hong Kong just reported 100 new H7N9 cases this morning. We are maybe sitting right on the pot right now. We don't know. So, I mean, I think that's the ones that worry us. The other ones surely can be problems. We can have worldwide, quote-unquote, epidemics, even to the extent of pandemic. We have a second one that is very different than all of these and kind of goes completely against what I just said, and that is the very slow-motion tsunami of antimicrobial resistance. That is a global pandemic unfolding that's going to take 20 to 30 years before it's fully realized. But when that finally happens, we are going to be on the edge of basically a pre-antibiotic era, where basically we will see a fundamental reordering of deaths. The AMR report just issued this past year from the group in England, a very thoughtful, comprehensive report, indicated that by 2050 at this rate, antibiotic-resistant infections will surpass both heart disease and cancer as leading causes of death. We'll be back to that age, and so I think that's a slow-moving one. but it's clearly the same phenomenon. I don't know. Andy, you...
No, I mean, I also have deep worries about antibiotic resistance. I mean, roughly, casting my eye around the room, half the people in the room will die of that. But probably not until you're much older. Usually the most likely place to get it is when you take it into hospital when you're older, or when you're not so old and you pick up a resistant strain, because the best place for these antibiotic resistance bacteria is hospitals, which is why those maps center around lots of the major medical centers in the world, because that's where these things are emerging. In terms of the other thing, I almost totally agree with you about the influenza. We should be really worried about that, although we do know the good thing about influenza is we nearly always, everyone gets it. So you've got some cross immunity to there, and that's sort of topped up all the time until it makes one of these shifts. And then my biggest worry, as we discussed last night, is if we had a radically different influenza, which we're not going to need to see 1917, 18 again, because the world was suddenly connected much more in 1917, 18 than it had ever been before. We're now highly connected, so we're spreading these things around all the time. The big worry is we haven't got the vaccine making capacity to make enough vaccine. If we had a new, like last year we had Zika at the same time as we had a big yellow fever epidemic in Africa. If we'd had a new influenza at the same time, we wouldn't have the capacity to make the vaccine to cover all those different areas, and that worries me. The one other class of respiratory things is still the morbilliviruses. And there, measles, rinderpest was the most devastating pandemic we ever saw, eradicating much of the wildlife, the ungulates in Africa. The two things that have appeared that are like that are Hendra virus and Nipah virus. They're very closely related. So they are pretty scary things. And they're scary because they're much more efficient types of HIV. They wipe out your B-cell memory and make you susceptible to everything you've ever had before. And something like that really, really scares me.
I'll add, Susan, in response to your question, one thought about the concept of emerging disease. So where we sit in developed countries, we want to predict the next pathogen that will emerge here, or more likely come here from another place that will be a tremendous public health threat. But at the same time, of course, we need to remember those infectious diseases that are not emerging, that plague human society around the world every day in profound ways that are very important. And I think it's interesting when we imagine something like climate change affecting where things emerge, or the dynamics of diseases in different places. Those don't have to be diseases we've never seen before. Some of them will be diseases with which we actually have a long-standing history, but we'll be surprised about how their dynamics change, or where they show up, or places where they have been eradicated and they resurge again. So I don't if we call those emerging diseases, or how we'll think about them as endemics and pandemics, but they might be stuff we're quite used to.
And just follow up on that, and this is an area that I think we in infectious diseases, and from the standpoint of the work you've done, need to have a great deal of humility. No one would have thought that an arthropod-borne infection, classic arthropod-borne infection like Zika, would also become a very efficient sexually transmitted disease. And suddenly, out of nowhere, we had both angles going. And we don't have a history of that. People have said this doesn't happen, kind of thing. I sit here today, and I've been heavily criticized for this, but I don't give a damn. When you get old, it doesn't matter anymore. My greatest fear is Ebola is still going as a respiratory transmitted pathogen. Ebola, Reston did it very efficiently in the book The Hot Zone. While that's not a human pathogen, at this point, it's getting closer and closer to one in terms of what we see the changes. We saw changes in the Ebola virus in the West African epidemic that concerned us greatly. I just had that exchange this morning with Gary Clovinger, who is one of the world's leader in the area of Ebola research. And if we could see very subtle changes in the virus, it would not at all prevent it from becoming a respiratory transmitted pathogen. And if that were to happen, that would, again, from a dynamic standpoint, as you pointed out, Andy, that would put a whole new perspective to Ebola. Ebola was bad enough, but if we had a respiratory transmitted Ebola, it would be substantial. And a lot of people don't want to go there, and they say, oh, that's just scaremongering. And yet we get caught time and time again not thinking the unimaginable that actually is imaginable, that we should have thought about, and that's how we get caught. We should never have been caught with Ebola in West Africa. Urbanized Ebola was just waiting to happen. Wait till it hits Kinshasa. So we shouldn't be surprised. So, right.
Hi, I just wanted to go back to one of the points you made in the Zika part, which which I thought was really interesting, and it touches on policy, and that is dividing resources between vector control versus vaccine.
Oh, is the microphone on? I think it. Can you
You just
turn
need
it
to get closer, Craig.
on? You're on now. You're on now.
I could sing.
Yeah.
All right. Give me the
Welcome,
green light.
everyone. I just wanted to go back to your point about the division of resources between vaccination versus vector control with Zika. But especially in light of your further conversations just now, the vectors don't just carry one thing. And you highlighted some of that if you're emphasizing too much just one disease at a time, the dynamics get much more complicated. And certainly it seems that vector control is huge for yellow fever, malaria, and these same mosquitoes can carry all these. And there's also scope, people have discussed, like with equine immune deficiency virus, that is a vector-borne lentivirus or FIV version in horses. So I think that the complexity is often overlooked in the hysteria of the latest emerging disease.
Right.
Yeah.
And the broader context of the overall overwhelming pressure of all these different things is really a key point.
No, I mean, just thinking more about Zika and thinking about malaria, the vector-borne diseases are very different from the directly borne diseases, particularly if they are immunizing in any way, because then the mosquito is your friend. It's actually, if whatever virus it's carrying doesn't kill you, it's going to immunize you, which is one way. So how do you change the average age at which people first infected? And the epidemic does that the best way. If it keeps going, it brings that age down really quickly. So if you can get it down to people who are 2-year-old kids, then that's fine, because A, they're unlikely to transmit it sexually, B, they'll be immunized for life then, and that immunity will be topped up at a very, very slow rate when the disease dies out. It's not an acceptable public health solution. You've got to be seen to be doing something. So knocking the mosquitoes back, if you could knock them back 99% or 99.9%, you might begin to have an effect that stops the epidemic. But similarly, developing a vaccine for something like that is going to be the way to go, because, it's something I'm starting to get my head around. These vector-borne immunizing disease is its own worst enemy. It quickly creates this herd immunity that's going to knock its abundance back orders of magnitude. So that means it has to have some ways of changing its immunogenetic properties all the time. And we're beginning to see that with malaria and the trypanosomes, that this constant ability to change your coat and not be recognized must be an adaptation to that transmission efficiency, which means that you hugely immunize the host population. So unless Zika has that, it's probably going to die down really, really quickly. Or is it going to be more like influenza and be able to mutate at such a rate? But it suggests there are massive constraints on being a vector-borne disease because of the immunity you create, which hits you twice, because you're either biting somebody who's already immune or not being transmitted to by somebody who's already immune. So your chance of finding a susceptible host plummets with a really efficient vector-borne pathogen. And I don't think people have really explored that enough. But it does put a different set, it puts a similar set of evolutionary pressures on the vector-borne diseases as we see with an influenza that's so massively efficient at transmission.
Yeah. I think the only challenge to that is that there really is a misunderstanding about mosquito-borne diseases, and I see it frequently repeated. A mosquito is not just a mosquito, is not just a mosquito. If you're dealing with Anopheles, the primary vector for malaria, you've got a mosquito that lives in a very different habitat. Their spread is very different. If you have Culex tarsalis and you're worried about West Nile transmission, birds play a huge role in moving that around, and as does the fact that, we've done studies showing Culex tarsalis can fly 20 miles in one night here in Minnesota on a good wind. Whereas if you have Aedes aegypti and Aedes albopictus, which is the primary vectors of the disease Zika we're talking about, dengue, they won't fly across a city road. They won't go across an open field. And you can actually localize their breeding, and you can do a lot to eliminate it, and you can clear literally small towns. We have the same problem here in Minnesota with a disease called lacrosse encephalitis, which we know they breed in tires, crotches of hardwood trees, etc. When we have a case, we can go in and survey that area and find all the breeding sites, eliminate them, and it never comes back again because, again, the mosquito has to be there. So with Zika, you could, in a sense, do what Gunnison, Colorado did in 1918, where they basically circled the entire city and nobody came in and out of the city for 12 months, and they never had a case of influenza. Okay? In a sense, you can kind of do that with the Zika at a very localized level. You can do that. But that's also the same problem. I have a problem with control of Zika, where they're talking about using genetically modified mosquitoes, et cetera, because you literally have to drop them every 100 feet practically into these areas where it didn't cross the road, whatever, to make it work, because they're not going to fly like Anopheles and Culex and actually cause that dynamic change that it would work with those mosquitoes. So I think one of the simplistic things has been just assuming a mosquito is a mosquito, and they are as different as people. Yes?
I was curious on the aspect of the ecology in which diseases are emerging. So, for example, human production systems have created very different ecologies in, so cattle growing in pens where there's lots of antibiotics, the SARS epidemic being driven by large poultry farms. So has that thread crept into the research of trying to really understand these new ecosystems that we've created as humans and the implications that those have for the ecology that the disease sees?
No, I mean, I think that opens a fascinating set of questions that I didn't really address. Looking at certainly, human change of the natural world is massively increasing contact rates between humans and more humans and different potential reservoir species that might cause things to spill over. Converting land from forest into agricultural land and changing the relative abundance of herbivores to carnivores and things that vectors can feed on also makes a huge difference to sort of disease emergence. People are beginning to look at that. Again, my slight concern of lots of it's correlative without many people actually doing experiments on it, but it's quite hard to do experiments on land use change, but not impossible. And then how do you make it predictive, or are you just mainly sort of assembling anecdotes? And we need more synthesis of that, and I think that would be as valuable an exercise as the sort of drawing maps from 200 data points exercise.
Do we have any web calls, Okay, we're good on that. We have time for one more question here. Anyone have a question? Go right ahead. Thank you. Mike.
My question was regarding, you talked about antibiotic resistance being a problem in the next 25 years. Are there any, is there any research going into like using mycological resources to combat those things, or maybe using CRISPR to make specified bacteria that could, probiotics or something that could fight a specific infection in a person?
Again, it's not really my area of expertise, but I talk with people about it. I mean, one possibility, it relates to the CRISPR thing, is many of the bacteria that we're worried about are attacked by phage. They used to be a whole part of Russian medicine that dealt with phage therapy, using phage to treat bacterial infections before antibiotics came along. So it may be that we have to go back and start looking at phage as a way of curing, of treating bacterial infections where the bacteria are resistant to the drugs we might use.
Yeah, I think it's going to be a comprehensive response. We're going to, first of all, have to learn to use a lot less antibiotic. So much of it's unnecessary. Last year, China alone exported 80,000 tons of antibiotics for use in animal farms around the world. We don't do much better in this country in humans and animals in many ways, and much of it's unnecessary. The second of all, besides antibiotic stewardship, is we obviously need to develop new antibiotics for which right now there's not a big market. If I'm a pharmaceutical company, why do I want to put money into a product? I don't want you to use it unless you absolutely have to, and then use the least amount you can. That's not at all a profit center like a drug you can take every day for a lifestyle issue. Finally, the third thing is, I would agree wholeheartedly with Andy, but I'd expand onto that and say this is where vaccines are going to become only more important. Today, if we could take care of some of the diarrheal pathogens of the developing world with vaccines, where we have unsafe water, we could greatly reduce the use of antibiotics that result in a lot of resistance. respiratory pathogens the same way. So it's going to be a multi-pronged approach. My last recommendation has been for some time, we need an intergovernmental panel on antimicrobial resistance like we have for climate change. And if we could bring the world together, because this is a worldwide issue. What happens in China, what happens in India, what happens in Africa, what happens in the Americas and Europe are all linked together. If we had the perfect system in the United States, we'd still have all the leakage of antimicrobial resistant bugs from around the world. So we really need an intergovernmental panel right now that can do what you and others have done in climate change and done the work there.
Can I add one more?
Sure,
thought
go
to
right
that?
ahead. You're going to make it right under the wire.
So this is an answer to the specific mechanistic approach, but stepping back and thinking about what is the phenomenon that causes antimicrobial resistance, it's an intensity of selection. And so whatever approach, I mean, this is, over application of antibiotics, and I wholeheartedly agree with you that the sort of economic model is not appropriate or doesn't incentivize those interventions that are effective and not over applied. And it's not just antibiotics, it's also pesticides, right? So over applying pesticides is the fastest way to get yourself a vector that needs a new pesticide, which I suppose is an economic model unto itself for chemical companies. But how you create a system by which a much more thoughtful approach is used and it isn't driven by being paid per application, but that fundamentally reduces the intensity of selection that is applied to the agent you are trying to control, will always be more effective.
Well, I want to thank both our speaker and coming from Princeton today, our commentator, for a really very interesting afternoon, and thank you very, very much. And this will bring our program to a close, and thank the audience for your questions, for your participation, and again, remind you, if you miss something you really want to desperately hear again, this is going to be put up. It's been a live cab, and I think it'll be posted within several days. So thank you very much, and have a good day.
Thank you.